is one good example of that. So even though it says it's about soybean seeds, you can think about biomedical applications and cancer pathology and stuff like that as well. So, Matthew is currently a Department of Defense Fellow here at the University of Illinois and working in Professor Rohit Bhargav's group. He has a BS in chemistry and a BA in biology from St. John Fisher College in Rochester. And he got his PhD in chemistry from Michigan, where he worked in Raman Spectroscopy. So we'll like you since you came here from Michigan. So don't go back. Matthew's won the several great awards, including the University of Michigan Distinguished Dissertation Award, the Thomas Hirschfeld Award in Applied Spectroscopy, and the Casimir Fejan Award in Chemistry. So welcome, Matthew, and look forward to your talk today.
Thank you. Non-conventional Raman spectroscopy applications towards the compositional analysis of soybean seeds. So soybeans are actually a pretty big commodity here in the United States and worldwide. In the U.S., 76 million acres of land are devoted to growing soybeans. And this translates into about a $40 billion industry, 23 billion of which are exports. Now, in any industry, the goal is really to gain the most value from your product that you can. And in soybean seeds, not all soybeans are created equal. Premiums are paid for nutritional content. So a lot of work has gone into trying to develop approaches to quantify various nutritional components of soybean seed. So the things that people are interested in, buyers are interested in protein and oil content, as well as various amino acids, various fatty acids, and various sugars. And the current approach to analyzing soybean in a non-destructive way is near-infrared spectroscopy. So near-infrared spectroscopy is a type of spectroscopy that's been around since the 70s. It's underwent quite a bit of development. Right now it's the current industry standard. Now, I'm a big believer in Raman spectroscopy, and I think Raman spectroscopy holds a lot of potential for quantifying these specific components. Currently, protein and oil is the only thing that NearIR can quantify to the industry needs, whereas Raman spectroscopy holds a lot of potential for amino acids, fatty acids, and specific nutrients like that. So in this talk today, I'm first going to go over an introduction to spectroscopy. Then I'll talk about how spectroscopy can give you chemical contrast. I'll follow that up with an instrument we built that demonstrates you could use Raman spectroscopy to quantify protein and oil content in single soybean seeds. And then I'll follow that up with an instrument we built for bulk soybean analysis. So what is spectroscopy? Spectroscopy is basically taking light, breaking it into its component colors, and trying to get information from those colors. So an example of how you get information from color, here's a picture of Waldo. Now, here, Waldo, is red, has a red shirt on with a red hat. But in this picture, you can't actually discern which one is Waldo. By adding color to this, you get information. Color gives you information. So here you can see Waldo's right there. So spectroscopy is converting light into color, light into information. This can be qualitative by observing what colors are present, or it can be quantitative by not only observing what colors are present, but also the intensity of those colors. Now, the human eye can actually only see 3 colors. It could see red, green, and blue. All the colors on the screen here are combinations of red pixels, green pixels, and blue pixels. Now, with scientific instrumentation, you can actually see a whole range of colors, a continuum of the electromagnetic spectrum, where the visible region is actually a very small portion. So for the work that, for the spectroscopy that we do on soybeans, the near-infrared spectrum is right in this range. You can actually gain additional information by looking maybe out into the mid-infrared or using other parts of the electromagnetic spectrum. So the typical spectroscopy experiment would look like this. You would take a range of colors with known intensities. You would introduce it to a sample, and you would collect the light that comes out of the sample. And you would then ratio the light that went in to the light that went out, and that would give you an idea of how the sample modifies the light, or how the light interacts with the sample. So a general definition of spectroscopy is the study of the interaction between radiation and an analyte as a function of wavelength. Now, if we were to do a spectroscopy experiment here on water, if you would take a whole range of wavelengths, so the ultraviolet all the way out to the far infrared, you could put that through water, And then you could ratio the light that went in to the light that went out, and you get a spectrum that looks like this. Now this is actually 4 different types of spectroscopy. This is UV vis spectroscopy, visible spectroscopy, NearIR spectroscopy, and IR spectroscopy. The method's the same. You ratio the light that went in to the light that went out, and ultimately the only thing that changed is the range of wavelengths that you put through your sample. So the range of wavelengths that you choose actually give you different chemical information. For example, UV visible spectroscopy is electronic spectroscopy. And what this does is light absorption is a result of electronic transition, so electrons moving from a ground state to a higher energy state. Infrared and Raman spectroscopy are vibrational spectroscopies. So what these are looking at groups of molecules and specific vibrations that are moving as a result of light incident on them. Now, Raman spectroscopy is different than the other types of spectroscopy I'm showing you here. With Raman spectroscopy, you use a single wavelength of light, and you put that single wavelength of light through your sample. So it's common to use something like a laser to get many photons at one wavelength and then put that through your sample. So for an illustrative example, I'm going to use 785 nanometers as our Raman excitation frequency. So if you think in terms of photons, when a photon interacts with a sample, a couple things can happen. Number one, it could be absorbed. So if it's IR light, the light would be absorbed, and that would promote some vibrational states. If it's UV or visible light, that can promote electronic transition states, and the light's absorbed. If the light's not absorbed, one of 3 things can happen. The light will be scattered through the sample. And how it scatters depends on the molecules that are present. So the first type of scattering is Rayleigh scattering. With Rayleigh scattering, what happens is a photon comes in, it interacts with the sample, it promotes molecules to some kind of virtual energy state, and then the same photon comes off. So a 785 nanometer photon comes in, and then the 785 nanometer photon leaves the sample. Second type of scattering is Stokes-Raman scattering. And with Stokes-Raman scattering, what happens is a 785 nanometer photon might come in, and then it leaves some of its energy with the sample, and that results in a lower energy photon coming out. So this might be an 800 nanometer photon. Alternatively, you could have anti-Stokes scattering, and this is where a 785 nanometer photon will come in. The molecules are already vibrating because they're at room temperature, so the photon can actually gain energy from the molecule, and as a result, you would have a higher energy photon coming out. So if you put a 785 nanometer photon in, something like a 700 nanometer photon could come out. So what this looks like in terms of instrumentation is you would have a photon come in, 785 nanometers, interacts with the molecules, and then in the Raman spectrometer, if 785 comes out, this is Rayleigh scattered light, that gets rejected by the Raman spectrometer. Alternatively, a 785 nanometer photon could come in, interacts with the molecules. This time you have anti-Stokes-Raman scattering, so it gains energy from the sample, and you have a 782 nanometer photon come out. This makes it through the Raman spectrometer and then gets sorted by wavelength. Alternatively, you have a 785 nanometer photon coming in, interacts with the molecules. This time it loses some of its energy to the molecules. As a result, you have a lower energy photon. That makes it through the Raman spectrometer, and it gets sorted by wavelength. Now in reality, you have a laser, so you have billions and billions of photons interacting with your sample. The vast majority of those photons are actually Rayleigh scattered, so you don't actually look at those. Those get rejected by the Raman spectrometer. The ones that are Raman scattered make it through and then get sorted by wavelength. So a Raman spectrum looks like this. It's basically a histogram of photon counts that's collected over time. So if you take this, put it on its edge, you end up with a Raman spectrum that looks like this. So on this axis, you have Raman intensity, which is photon counts. And on this axis, you have a shift from an excitation frequency. So if we excite at 785 nanometers, you have an 810 nanometers to 915 nanometers. And this is generally considered the Raman fingerprint region. And it's usually reported in terms of wave number shift in order to compare different laser wavelengths if you excite with a different laser other than 785. So that is an introduction to spectroscopy and how Raman spectroscopy is different than the other types of absorption spectroscopy that are out there. Now, spectroscopy gives you contrast. So to compare the different types of contrast, I got a couple slides that just compare NearIR to IR to Raman. So with NearIR spectroscopy, the light tends to scatter more than it's absorbed, so you have a greater penetration depth. And this is the reason that NearIR is commonly used in quality control applications for the agricultural industry and soybeans as well. Because the light scatters more than it's absorbed, it can go through very thick path lengths, and as a result, you can get the light through an entire soybean or through an entire apple, something along those lines. This spectral information arises from broad spectral overtones as a result, and as a result, is it difficult to assign spectral features to specific chemical components. So the chemical information you're achieving here is basically groups of molecules moving together, and as a result, you don't know anything about the specific vibrations that are present. So they're spectral overtones. Light scattering tends to be a problem, so corrections are needed. So spectroscopy tends to follow Beer's law in order to do quantitative things. And in Beer's law, you need to know the sample path length. So because the light scatters, it doesn't go directly through the sample, so you don't really know the path length. There's a lot of very good corrections for scattering regimes that actually work pretty well in the NearIR. But this is something to consider if you're doing NearIR spectroscopy. IR spectroscopy, light tends to absorb more than it's scattered, and as a result you have very little penetration depth. And for this reason, in order to do IR spectroscopy, you have to work on very, very thin samples. Samples tend to be on the order of maybe 5 to 10 microns thick. The spectral information arises from specific vibrations arising from molecular functional groups. As a result, you can assign spectral features to specific chemical components. So the chemical specificity in IR spectroscopy is superior to that of NearIR spectroscopy. Sample thickness and preparation tends to be the biggest difficulty with IR spectroscopy. So if you want to take IR spectrum of something like tissue, you have to microtome it to fairly thin sections. And you also have to make sure there's not a lot of water present, because water has a very large absorption in the infrared regions. Now, Raman spectroscopy is actually the best of both worlds. With Raman, you can use any wavelength you want. So you can choose a 785 nanometer laser, a NearIR laser, that achieves the penetration depth that you gain with NearIR spectroscopy. But at the same time, it's also a vibrational type of spectroscopy that gives you similar chemical information to what you achieve with mid-IR spectroscopy. So you have very high chemical specificity in a Raman spectrum. Now, the quantum efficiency of Raman is low, and that's why it hasn't really been developed for things like agricultural applications where you're analyzing whole soybeans. Quantum efficiency being low means it's a low light event. You need very, very sensitive detectors. You need very high-powered lasers in order to get nice Raman spectra. And the reason that it's actually applicable in the research I'm showing you here is because detectors, lasers, filters are becoming better and better each year. And as a result, we're able to do things that we couldn't have done 20 years ago. So all spectroscopy follows Beer's law. So I'm going to show you some calibration curves in future slides here. And these calibration curves are based on this principle, where you have a concentration. So you can get the concentration using wet chemistry approaches. And you have basically a known amount of a sample that you're looking at. And then what you do is you take a spectrum of that, and that spectrum gives you a response. And this response is going to be linear. So then what you could do is use this linear relationship. So you get an equation for this line, and you can take a spectrum, follow that over to the line, and then figure out what the concentration is. So that's how you quantify using both NearIR, mid-IR, and Raman spectroscopy. Now, conventional Raman spectroscopy, as well as mid-IR, and not so much NearIR, you can do spectral imaging. So spectral imaging is basically taking, getting your spatial resolution using point mapping, and then getting a spectral resolution by taking a spectrum at each one of these points. So you get your X, your Y, and then your Z-axis is going to be a spectrum. Okay? So in conventional Raman spectroscopy, what happens is the light would come in, it goes through a lens, it focuses to a point, the Raman spectra is generated, the Raman signal scatters in all directions, that gets collected by the lens and then goes back up to a Raman spectrograph. Now by taking multiple points over time, you get a spectrum at each one of these points here, and you can put together a map of Raman spectra that looks like a Raman image that you see here. And in order to visualize the distribution of chemistry in this image, you could pick a couple spectral bands. And I chose red, green, and blue because those are the colors your eyes are sensitive to. And this gives you an idea of the distribution of chemistry within the soybean sample. So if you compare the three different types of spectroscopy, you have NearIR here. You can kind of see the distribution of chemistry shown by the NearIR spectrum. Here you have a mid-IR spectrum, and you can see there's a lot more information here. There's a different distribution than what you're seeing in the NearIR. And then here's a Raman image of the same soybean section. And you can see there's pockets of, I guess, teal here and orange here. Those correspond to sugars. The purple here and the purple here correspond to oils, and the red might correspond to protein and various different chemistries. So understanding the relationship between what spectral bands you observe in the Raman spectrum and the image, you have to kind of look at known standards in order to figure out what you're actually visualizing in this distribution. So using the mid-IR spectra or imaging, we can visualize the distribution of protein and oil in soybean sections, and we see that the distribution is actually fairly heterogeneous. It's not a uniform distribution of protein, and these are 4 different soybean sections that we took infrared images on. You can see this soybean section, which has a protein content of about 50%, has these splotches of protein here, whereas this one that has a protein content of 51% has actually large pockets of protein, whereas the other portions of the seed are low in concentration. So what this means is if you use conventional Raman spectroscopy to develop a calibration model, if you took just a point spectra here, you would get a very different calibration than if you took a point spectra here. So you need to take representative sampling of the soybean in order to actually develop a proper calibration model. So in order to do that, we turn to actually transmission Raman spectroscopy. So conventional Raman spectroscopy, if you take a point sample on an entire soybean, what you would get is a signal that's actually surface weighted. So we have a pharmaceutical capsule here that has a gel cap, and then inside is a powder that has pharmaceutical ingredients. And if you take conventional Raman spectra, what you end up seeing is basically just the gel cap. This is a fluorescence signal that arises from this gel cap. If you take a spatially offset Raman measurement, so this is different than the conventional Raman approach, what you would do is you would illuminate in one position and then collect spatially offset from that. And the idea is that because this sample is light scattering, the photons would diffuse through the sample, and then if you collect offset from that, you would get some kind of representative sampling from the path that the photons took in the diffusion process. And through this approach, you end up getting a spectra that looks like this. So you actually do get some of the active pharmaceutical ingredient in this Raman spectrum. Now, if you take a transmission approach, so you basically take the illumination and bring it down to the bottom, you end up getting a much better representative sampling of this pharmaceutical. So that spectra looks like this. So taking a similar approach with soybeans, the idea was we could take a representative sampling of the soybean and then get a representative spectrum that we could develop calibration models on. So this is actually a general problem. Like we apply it to soybeans, but it could be applied to process analytical technology, where you might be looking at fluids mixing through tubing. You could use it for biomedical applications, where we've actually used it to recover bone spectra through the skin. You could use it in pharmaceuticals, where you want to look at the distribution of active pharmaceutical ingredient. Or we're using it in soybeans here, where we're trying to quantify economically and nutritionally valuable components of soybeans. So light scattering it looks like this. If you have no light scattering, what I have here is a beaker full of water, and I have just the Illinois emblem inside the beaker. And if you increase the scattering, it's pretty clear that you can no longer actually see this eye emblem. And the reason for that is because the light, as it travels through the beaker, ends up spreading out, diffusing, and you can't actually image anything inside of that without doing some kind of math on it. And then this is what it looks like in a cuvette. you have a cuvette full of water, and then if you increase the scattering, you can see the laser beam profile spreads out, diffuses as the scattering levels increase. Now, the reason this is important for Raman is you're only going to get a Raman signal where the where the excitation photons actually propagate to. So in highly scattering systems, you can see the laser actually spreads out and propagates pretty significantly, which means if this was a soybean, which is highly light scattering, you would get laser light kind of spread out throughout the entire soybean. And then there is some Monte Carlo modeling that basically demonstrates that you get representative sampling in a transmission approach. So here we have a light scattering sample, and what we're showing is a histogram of where the Raman photons actually come from. Okay, so the single soybean instrument. Okay, so on that last slide, I showed a Monte Carlo simulation. We actually did this for soybeans. So here we took a CT scan of the soybean in order to figure out what the internal structure of the soybean looks like. We then generated a computer mesh so we can model photons propagating through the soybean. We labeled this with the proper absorption and scattering coefficients. We then illuminated a soybean on one side, and we just watched the photons randomly walk through the soybean, and had a detector that had a certain numerical aperture, and we figured out where the Raman photons originated that we actually detected. And if you look at a histogram of this, so this cross section through the sample here, you end up seeing something like this. So you end up getting photons largely concentrated in here, but you also get some from the surrounding area too. And this is center weighted as opposed to being surface weighted. So if you look at a soybean actually has this outer skin on the outside that isn't useful. So you would want to get all the information from the interior of the soybean and not the exterior of the soybean, which is what conventional Raman spectroscopy would give you. Then I got an image here of a soybean that we illuminated with 785 nanometer light. This is 200 milliwatts. And you can just see it's kind of glowing red. And this just kind of supports the idea that the photons just diffuse and bounce around throughout the entire soybean. OK, so we wanted to see, can we actually collect a Raman signal from the soybean? So in our first set of experiments, we had a 785 nanometer laser that was focused onto the back surface of a soybean. And then we just collected all the light that came out of it. And we ended up seeing spectra that looked like this. Now, each one of these spectra took an hour to collect, and that's largely because of the inefficiency of our setup, and this was only a 40 milliwatt laser. But we get a definite Raman spectrum from 4 different types of soybeans. So you get a phenylalanine band here, which corresponds to protein as well as the amino acid phenylalanine. You get CH2 bands, and you get Raman spectra. This is a Raman spectra that is recognizable. But the acquisition time was quite long. So based on that data and some of the simulations that we did, we were able to apply for funding, and we got funded to put together an instrument that could measure Raman spectra from single soybeans efficiently. So this is the instrument we ended up building. And what you have here is you have a sampling stage, and the sampling stage consists of a series of holes that you would put the soybeans into. And then there's a hole that passes through this plate. The laser light comes from the bottom, illuminates the bottom of the soybean. This is a collection lens that is automated. It moves up and down and focuses onto the soybean. Then that light gets collimated from the sample, goes to fiber optics here, which brings it down to the Raman spectrometer. So this thing's automated. It's controlled by LabVIEW. We're able to load it up with soybeans, set whatever parameters, and then come back maybe the next morning, and all our data is stored in the computer. So with this, the spectra that we acquire with this instrument looks like this. This is a hyperspectral system, which means that we collect about 256 spectra at one time. The CCD frame would look like this, where this is the spectral axis shown in pixels, and this is the slit height, which basically shows the different spectra we collect. If you take this and put it on its side, you get a spectrum. You get your 256 Raman spectra look like this. Now, to develop a calibration model for one soybean, we only need one spectra. So basically, in order to improve signal to noise, we just bin these spectra and get a single spectrum. So acquisition time was one thing we wanted to look at. We wanted to take good data, but at the same time do it as quick as possible. And for this study, what we did is we changed our acquisition time from 2 minutes up to about 10 minutes. And you can see the different spectra corresponding with those measurements here. So this is the 2-minute measurement, this is the 10-minute measurement. And we did a signal to noise comparison. And we find that after about 5 minutes, there actually is no improvement in signal to noise. And this is largely because we're gaining noise from the readout of the CCD. But if you look at this, the 2-minute measurement actually looks pretty good. So just to compare those different measurement times, we developed a leave one out cross-validation calibration model. And what I'm showing you here is a correlation plot. So I just wanted to explain what a correlation plot is. A correlation plot, you take wet chemistry. So these are reference values where you take a soybean, you grind it up, and you're able to get ground truths for protein and oil content. And then this is the spectral response predicted by the calibration model. So in order to figure out how well you're doing, what you want to do is you want to take one of these points, see what the wet chemistry said, see what the calibration model said, and if it matches up perfectly, you're going to be along this x equals y line right here. So for our instrument, the different acquisition times, we took a 2-minute measurement up to 10-minute measurement. You can see that the root mean standard error, so this is basically the actual minus predicted, is about the same, statistically the same for all 10 measurements. So this means that we basically took our calibration and validation set for the next set of samples at a 2-minute acquisition time. Now, with the current instrument, and even with the instrument in this configuration, we probably could get down to maybe a 30-second acquisition time. But this is all reasonable for what the people that we're working with want to do with this instrument. So the 30-second acquisition time is actually pretty good. So we developed a calibration model where we did 236 soybeans for oil and 237 soybeans for protein. And this is what our calibration model looked like compared to the wet chemistry. What you see in the green, the yellow, and the red is the 1 standard deviation. The green is 1 standard deviation in the wet chemistry, so that's basically the error of the reference method. And our calibration model is within those error bounds. And this is the validation set. So we validated on a slightly larger set. So we had about 300 soybeans for oil and 300 soybeans for protein in the validation. And you can see we're doing pretty well. So if given a soybean, we could predict the protein or oil content within about a 1% standard error. So that's not bad. And this is basically about the same as what NearIR could do. Now this is actually the first demonstration of using Raman spectroscopy to quantify non-destructively the protein and oil content of soybean seeds. So that's fairly significant. Now NearIR again is the industry standard, and that actually is used to quantify protein and oil content. Now what NearIR has trouble with is amino acids, fatty acids, looking at specific components. So in order to tackle that, we moved to a bulk soybean instrument. The reason we went to a bulk soybean instrument is because if you look at the relative contribution from the various amino acids, these things add up to make your protein content, and the fatty acids add up to make your oil content. So the concentrations of the amino acids are much smaller than they are than protein or oil. So as a result, you have to actually do the reference method, the wet chemistry, on bulk samples. So in order to compare, we had to take bulk soybean measurements. So we went from the single soybean to the bulk. So we designed an instrument in AutoCAD, and also used some ZMax to do some optical modeling. And the instrument looks like this. It has a hopper on the top where we can just basically pour a bunch of soybeans into. It has a little turn screw that feeds the soybeans from the hopper into a sample chamber here. So there's some soybeans in the sample chamber. The optical path goes like this. After the measurement's collected, there's a trap door here that opens up and drops the soybeans into this box here, and then we can just repeat the measurement and do multiple samples with a specific variety of soybean that fill the hopper there. So the optical path looks something like this. We have a laser beam come in. We collimate the laser beam to about a 2-inch waist. That collimated light goes through the samples. We collect the samples with a lenslet array, and that lenslet array then focuses the light onto collection fibers, and this is a hyperspectral measurement as well. This is actually the same spectrometer and set up from this side over that was used for the single soybean instrument. So in our labs, we basically have both of these on a table, and we just flip a couple mirrors, and we're able to use the same spectrometer for both instruments. Now the lens array is actually kind of a neat way for solving a problem. We want to collect a light over a large surface area, and at the same time have high efficiency, high numerical aperture. So the way to do this is to have multiple focuses that collect a lot of light, so higher NA lenses, and then we take all that light that's collimated from the lenses and then focus those into the fiber. So this is actually fairly efficient. Now the one problem with this was that each one of these spots is fixed. So in order to, you can imagine if the sample loaded into the soybeans loaded into the sampling chamber differently each time, and one of these focuses was like in between two soybeans, that would cause a problem. To solve that, we basically made this rotating lenslet array. So now over the acquisition time, which could be anywhere from 30 seconds to maybe 2 minutes, the signal basically gets averaged, and as a result, you get signal from 2 concentric rings. And just to kind of show you what that looks like, I just put laser through the other side, and you can see how the collection spots move as a function of time. So the Raman spectra we're able to obtain with this instrument looks like this. It's pretty good. This is about 12.5 millimeter path length. And in the experiment, in order to test out this instrument and see how good we can collect specific amino acids and fatty acids, we had 52 soybean varieties that are representative of the different soybeans that grow around Illinois. We had 300 milliwatts of laser power. We did 5 runs, and each run was 10 minutes. So for a given variety, we had 5 replicates basically, and each replicate took 10 minutes to acquire. And then we did the wet chemistry for a couple specific amino acids and a couple fatty acids that people are interested in. And these are basically the results for that. So here we have fatty acids. So these are some specific fatty acids that animal feed people are interested in. We developed the calibration model using the open circles, and then the validation models on an independent set of 26 soybean varieties are shown in the closed circles here. And then for amino acids, we're also doing very well here. So again, this is actually the first example of using Raman spectroscopy to predict fatty acid and amino acid content. And based on stuff we looked up in the literature, this is actually quite a bit better than what NearIR has been able to do in the same application. So one thing I will mention is these acquisition times were 10 minutes per run, but we're only using 300 milliwatts of laser power. Since this study, we've actually upped our laser power to about 3.5 watts. And because we're not focusing the light down, we're using a collimated beam, we're not actually burning any sample at that power. And we get much faster acquisition time. So we're at about 40 seconds for the same signal and noise that you're seeing here. Instead of 10 minutes per run, it's about 40 seconds per run, which is pretty good. The industrial folks would like to see it a little bit faster, but I think we have room to even improve above and beyond 3.5 watts. We can go up to 5 watts right now with a commercially available laser. Okay, so in summary, I gave you an introduction to spectroscopy and showed you how Raman spectroscopy is slightly different than the absorption approaches to spectroscopy. I compared the different chemical information you get from the 3 methods. I showed you our single soybean instrument and our bulk soybean instrument. So with that, thank you for your time. I could take questions.
Picture where you were actually imaging within a beam, right?
Yeah, so this was, which
early on, you showed pictures that looked like beams and you were imaging and different components. And I'm just wondering, why, is that an important piece of information, the distribution, say, within? Or is it just bulk information that's all that's important? So
depends who you talk to. Plant breeders are very interested in looking at the distribution of various amino acids. And one thing that is of interest is apparently as a soybean plant grows, different environmental, even if the soybean had the same genetics, Environmental influences affect what amino acids actually get deposited, and how they get deposited, and how they accumulate. And apparently sometimes amino acids that are there during one stage of seed development disappear later on in the seed development. So if this is an amino acid that you're interested in, maybe it makes more sense to harvest the plant sooner than later. So that's one thing that's of interest. And that's where the distribution comes into play, yeah.
Okay. Does this information, could this affect things like price? Oh,
absolutely, yeah. So knowing, basically anytime you know what you sell, you can get more money for it, if that's something that people are interested in. So being able to quantify the amount of lysine in a soybean, you can actually sell that to people that formulate animal feed, and then they know what they're putting into their animal feed, so then they can turn around and sell it to their customers at a higher price. Yep. Yes, sir.
So I know you said that you're using, you occasionally said how much power you're using for each setup. Is there a limit on like how much you can do? Because I know you said these methods are non-destructive and that you're not cutting up the soybeans really. But how much light can you put into it until something happens, and have you reached that limit?
So on the single soybean instrument, we actually focus down the light in the first iteration of that, and that's kind of unnecessary in anything that's a light scattering approach. So when you focus down the light, you have higher fluence, and fluence is basically the power per unit area. And that could cause burning. So even at NearIR wavelengths, there is some absorption. And if you have more photons coming into contact at the same spot, the light that absorbs would basically cause increases in temperature, which could cause burning. So in applications where you're focusing the beam down, the power actually matters quite a bit because you don't want to destroy your sample. In the bulk soybean instrument, we have collimated light hitting our sample chamber. And that collimated light, I mean, you can put that up to pretty much as high as you can buy a laser that would go up to that. Right now, we got a laser that was 3.5 watts, which is a pretty powerful laser. The strongest one on the market that I know of is 5 watts, and that's increasing every day. But a 5-watt laser is pretty powerful.
Yeah, because I know with lasers, especially, you say, oh, it's only 5 watts. It's it's 1/60th of what comes into most, well, not 1/60, it's a fraction of what comes into most appliances. For a laser, like, it's a lot. So
because a laser is coherent,
yeah,
and it's also monochromatic. So like light bulbs, for example, give a broad spectrum of light. So you couldn't necessarily like take a filter and get that much. And not only that, with light bulbs, you can't collimate the light because it's not coming from a small point. So collimating stuff with extended sources like light bulbs. Yeah. Okay.
All right,
cool. Sure.